Hello learners, I am Dr. Rekha Sharma, Associate Professor, UGC Human Resource Development Center, Rashtrasant Tukaroji Maharaj, Nagpur University, Nagpur. Today we will study glucosinolates. Glucosinolates are a large group of sulfur containing glucosides present in cruciferous vegetables belonging to Brassicaceae family, cabbage, broccoli, cauliflower, mustards, rapeseed, canola, etc. are the examples of cruciferous vegetables. These vegetables are said to be protective against cancers of lungs and alimentary tract. These compounds remain intact unless brought into contact with enzyme myrosinase by pests, food processing or chewing. The hydrolysis of glucosinolates form various metabolites like isothiocyanates, nitriles, oxaloidin, 2-thiones and indole-3 carbinols. These are chemically reactive and biologically active compounds and have been found to be effective in reducing the risk of developing chronic diseases. Glucosinolates can be gained or lost by vegetables during storage. They may be degraded or leached during processing or preserved by thermal inactivation of myrosinase. The bioavailability of the glucosinolates and their breakdown products depend on storage, time, temperature and packaging as well as inactivation process of myrosinase enzyme. The popularity of Brassica products is increasing because of their nutritional value, anti-cancer, antioxidant and anti-inflammatory properties. Now in this section we shall study the definition, structure and hydrolysis and sources of glucosinolates. Glucosinolates are any of various bitter sulfur containing glycosides found specially in cruciferous plants such as broccoli, cabbage and mustard that when hydrolyzed form bioactive compounds such as isothionates, including some which are anti-carcinogenic. Glucosinolates are a group of secondary metabolites synthesized by plants. They contain sulfur groups and are mainly found in cruciferous plants of Brassicaceae family. The plants in Brassicaceae family contain several bioactive chemicals such as glucosinolates, other sulfur containing compounds and erucic acid that can act as beneficial or harmful ways. Approximately 130 different glucosinolates have been identified in the botanical system, and glucosinolates have also antioxidant, anti-cancer, fungicide and bactericidal properties. Now let us come down to chemistry. The basic structure of glucosinolates include beta D-thioglucose group, sulfonated oxime group and variable side chain derived from amino acids. The side chain represented by R is derived from one of 8 amino acids and can be aliphatic, that is alanine, leucine, isoleucine, methionine or valine, may be aromatic, for example may be phenylalanine or tyrosine, or indole, that is tryptophan, or indole glucosinoids are associated with bitter flavors. A small change in the side chain leads to the formation of different glucosinolates. More than 200 side groups have been identified and cited in the literature. The general structure of glucosinolate has been shown in the figure. Now let us see sources. Brassica oleracea species include vegetables such as kale, collard, cabbage, savoy cabbage, Brussels sprouts, cauliflower, broccoli, kohlrabi and Chinese kale, while in Brassica rapa includes turnip, Chinese cabbage, pak choi, komatsuna. Other Brassica species include mustard greens, Chinese broccoli, Japanese mustard, gaijon, horseradish, wasabi, daikon, arugula, watercress and radish. Brassica vegetables are rich in sulfur containing compounds which are responsible for the rich glucosinolate content, the pungent smells and the spicy flavor. About 1500 to 2000 microgram per gram of glucosinolates are present in brassica vegetables and especially high in Brussels cabbage and broccoli. Concentrations in roots and seeds can be higher, up to 30,000 parts per million in horseradish root and 60,000 parts per million in mustard seeds. Hydrolysis of glucosinolates. Glucosinolates are relatively stable in plant cells. However, when the plant tissues containing glucosinolates is damaged, as in the case of preparation, that is cutting, chopping, mixing or chewing food, a beta thioglucosidase called myrosinase is released. The enzyme is normally stored separately from glucosinolates and located into vacuoles in a specialized cell type named myrosin cells. The hydrolysis of glucosinolate by myrosinase produces a molecule of beta D glucose and an unstable aglycone, thiohydroxymate O sulfonate. Spontaneous reorganization of this intermediate chemical rearrangement of leucine result in the release of sulfate ion and in the formation of metabolites, the structure of which depend on the nature of side chain R of glucosinolate and the physicochemical conditions of the medium. These may be isothiocyanates, nitriles, sulfides, thiocyanates, etc. Of the hydrolytic products of glucosinolates, glucoraphanin, gluconastrelin and glucobrasicin shows anti-carcinogenic activity. Furthermore, indole 3-carbinol, a metabolite of glucobrasicin, has inhibitory effects on human breast and ovarian cancers. Now let us conclude. So far we have studied the chemistry and hydrolysis of glucosinolates and their sources.